at the University of North Carolina, Chapel Hill, which is a beautiful place. And then postdoc at Duke, I think he was postdoc at Duke before going to Vanderbilt. And then from Vanderbilt, he moved together with Mark Anderson to the University of Iowa, where we were partners on the Duke Foundation Award that research chem kinase. And then he got an offer he couldn't refuse from the university, the Ohio State University, where he's still there. And I'll read you the title because it's long, I can't remember it all, but it says Peter Mohler, Professor and Director of the Dorothy M. Davis Heart and Lung Research Institute, Director of the Institute, Chair and Professor of Physiology and Cell Biology, and also the Associate Dean for Basic Sciences at the Ohio State University. So Peter has done some beautiful work on things that surround ion channels, that is ion channel regulation, ankyrin. You'll hear from him. Why should I waste my time? Okay.
So again, it's good to see old friends and new faces, and it's been a great last 24 hours at WashU, and unbelievable the amount of building that has been happening here, and not only here on this campus, but I had an opportunity to drive through the medical campus this morning, and it's amazing. It's quite amazing. So congratulations on what must be a large endowment and a lot of recruiting to do. So today I'm going to talk about two stories. One, I think a couple of you have heard a little bit about this story, but I think exemplifies what we're trying to put together in a sort of a WashU-like model where we bring biomedical engineers, with cardiologists, with surgeons to answer questions that obviously impact the clinic. And then the second part of the talk, be a little bit more basic and talk about some of the molecular mechanisms of some of the work that we've been doing for the last 7 or 8 years. So before I do, I want to thank a lot of the people that obviously did the work, a lot of grad students and fellows, cardiologists, just like what happens in your own lab. You know, you get nice stories where the 3 people in the middle met at lab benches, and 2 of them got married, and the 3rd one married them. So a lot of great things in science beyond just doing science, and again, this all happened because of this group. So I'm going to start with a story that a couple of you heard about a patient. This is a patient that this was the day he got married. Nothing wrong with this patient other than the fact that he was an attorney. But other than that, no cardiac phenotypes today. This was this patient 6 days later after experiencing sudden cardiac arrest. He was shocked 6 times, spent the next 30 days in a hypothermic coma where to preserve brain functions. During this time, he was outfitted with a defibrillator, and fortunately, he was, because over the next year and a half, he was shocked about 230 times, had PVCs basically constantly, shocked 90 times in one day. Unfortunately, like a lot of the patients that are seen here at WashU, resistant to conventional antiarrhythmic medications, he had ablation therapy at multiple institutions with all the fancy bells and whistles, and did not resolve phenotypes. This is a patient that can't work, he can't drive. He's had PVCs so many times that he's dropped his child going up the stairs. So not a lot that we could do with this patient. A deep dive into medical history revealed a mother with syncope that died suddenly in her 50s, and obviously this might be a candidate for what everybody wants to do now, which is genetic testing. You find a smoking gun, and everything goes well. Unfortunately, like a lot of the patients that are seen around the world, that this patient was known for negative for known Arrhythmia-G mutations. Looking at his EKG, he had a unique clinical presentation that really mimicked earlier polarization disease. And just for the bottom, so we can move on, that this was a patient that had, again, severe forms of ventricular fibrillation, even in the presence of quinidine here at the bottom. So again, a severe clinical presentation, which oddly only happened when he was about 38 years old. So this is kind of where we were about 2.5 years ago. multiple ablations did not resolve the
Where
phenotypes.
the ECG was in what leads? Can I just look at
Yes,
it? It's V1, V2. Okay.
So this is a patient, again, non-responsive to basically the Sigma catalog and not a candidate for transplant due to some other reasons that we could talk about later. So the question for this this patient, what was next? So we chose, in this case, to do whole exome sequencing, and everyone here has seen these slides where the price of sequencing has gone to the point where in certain cases it is appropriate or at least cost effective to have this as the next step. Again, working with our genetic counselors in the Arrhythmia clinic. What we learned, which is a lot of what some of you have seen that work in genetics, is that you have to be careful what you wish for. And these patients have just literally thousands upon thousands of variants that good luck finding anything. So graduate students were really the key. As much as we want to use supercomputers and computer-based learning, that really took graduate students sitting down and going over sequences over and over and over again to find potential causal or at least associated variants. And this is one of the ones that we really worked on a lot, which is a variant that was found in chromosome 7 in what we thought was a big sort of deserted sequence between two parts of a large gene. This is just the variant here on the bottom. When we looked at this sequence, though, which was supposed to be non-coding, it turned out that this variant lied within a region that actually, and this is just this coding region in this larger region, had a very large, again, reading frame that had not been picked up by the normal databases. And so the question was sort of twofold. One, what is this big chunk of protein doing? And the second is how does this function in this chunk of protein potentially associate with severe forms of cardiac arrhythmia? And so, again, I'm going to say a long story and try to make it as short as I can. But it turned out that this coding region we found was actually an alternatively spliced exon of a protein that's called DPP6, which I know Jean in her lab have studied as an associated protein for potassium channels, just to give a sequence here on the top. And this is, in fact, where the variant was in this protein. What we've learned since then is that unlike DPP6, which again is a long protein that crosses the membrane, that this new isoform with exon 5B actually has a stop codon at its C-terminus, and so it ends up with a truncated protein, and the amino acid here, which is mutated in this individual, is called H322R. So again, the question for a lot of us was, this is an area of biology where there's been a lot of questions of what this protein is doing, and in fact, for our lab, what is this new version of the protein actually doing? And so, again, very similar to what you guys have done here at WashU, is we've been developing repositories of human hearts. We can actually go immediately back and test to see whether these isoforms are truly in human LV, and it turns out when we look at this in human LV or RV, there's a lot of this protein. Well, there's a lot of the message there. Questions of whether the protein is there were resolved by making a new antibody that recognized this isoform. And again, this doesn't tell us about the levels, but it tells us that in fact this truncated isoform was present in the heart. Okay, so then the question was, what is the effect of this protein, and how does the mutation or the variant in fact alter the association with typical potassium channels? And this is work done by Faith Klein in the lab that allowed us to see that, in fact, that unlike what we hypothesized, which is usually true, that we found out that this human mutation actually increased the association of this small isoform of DPP6 with Kb4.3 in at least in these binding assays. The question, though, again, is what happens in real cells, and so we started off with HEK293 cells, and these are just looking at ITO and showing you that with potassium channel alone, this is 4.3 by itself, we see a current, and that this current is increased by co-expression with this new short isoform of DPP6T. The question then is what happens with the mutation, and this is just basically the same graph. You can see that the scale has changed, but we see, depending on how we express this and the amount of another protein called K-chip that we also work with, it actually increases the amount of current in the HEK293 cells by about 6 foot. Okay, so the question then is, we have this patient that's still in the hospital. This patient now is really starting to have additional issues. He's septic at this point. He's gone into a coma, and so is there ways that we can actually translate some of this work that we've done in the HEX cell to start thinking about how we can treat this patient in the clinic? And so a lot of you know Tom Hund, who did a lot of his work and trained with Joram and then was with Rick and worked with Catherine. So we gave this data to Tom and had Tom use his mathematical model of the human ventricle to be able to see what would the potential changes be in the action potential, and how could we potentially model this to come up with therapies for this patient. And so Tom was able to use his models to look at this, not only in the endo and the epi, but also to come up with potential ways to actually go in and treat this. And Tom came up with 2 different ways. One was, obviously, since ITO is a slowly activating potassium channel, is to increase atrial pacing. Unfortunately, this is a patient that didn't have an atrial lead. The second is to obviously block a lot of your ITO, or name your favorite potassium channel. Again, for the heart, no potential FDA-approved therapies for this were available. So again, this patient was at the point of really no return, and so we started looking through the literature with Cynthia Carnes, who's in the College of Pharmacy at Ohio State. And there was a compound that all of us are quite familiar with from the last 60 years of working in the lab, which is 4AP. And there's been some studies by a group called Acorda Therapeutics for use of this for the treatment of certain forms of multiple sclerosis. So we were able to get permission to try this therapy on the patient. So this is a dual therapy where we put this FDA-approved dalfampramine, the 4AP compound, as well as cilostazol, which would actually increase heart rate, and give it to the patient. And this is what you see with EKG and with this dual combination of treatment. So this is the EKGs before treatment, and then EKGs after the treatment. So this was obviously a point in time, and this actually makes a nice figure. The question, though, is did this patient have arrhythmia phenotypes resolved over months and weeks and years? So this looks now at the next 71 months of this individual's life. This was before we started working on the patient here on the left, and you can see over the months, following his initial presentation, again, literally hundreds of arrhythmia events and ICD shocks after he took this combination therapy, which obviously increased his atrial pacing rate, as well as taking the potassium channel blocker. The number of events went down to almost 0, and in fact, the only 2 events that they've had ICD discharges have been when this patient has been non-compliant with this therapy. So this is now at 71 months. This is an individual that is now out of the hospital. He's now back at work. They've had another kid, and he's kind of gotten his life back. So really what we were trying to figure out is, what is the impact of this variant, and in fact this new form of this potassium channel regulatory protein simply beyond this one patient in Central Ohio. There's been a lot of work on this same molecule by groups in Europe, where they've also found a very similar clinical presentation of arrhythmia and in fact sudden cardiac death in the mid-30s, the early 40s, also associated with variants in the promoter region of this same DPP6. Some of the questions that we're still asking in this story, one is, what is the reason for the presentation of this disease so late in life? It doesn't necessarily fit with anything we normally think about in evolution of something coming on so late. And the question that we're still asking is what's the role of DPP6 in regulation of this potassium channel versus the other 63 potassium channels that we know it can associate with. The second question really is the effect of these compounds on this patient, true and unrelated to this DPP6. Certainly there's a lot of other variants that could be causing it. What we're also looking at is sort of what is the impact of this sort of approach. Is it reasonable to think we can start looking at genetics in individual patients and broadly think that we can do this at the population level to make a difference in our healthcare system? And this is just sort of a slide to put this in a little bit of a context, to think about the idea of using genetics or using imaging to go after high-risk populations. And a lot of you have seen these data, but it's actually interesting to go through a little bit. And we now know that about 1% of our population, at least in the United States, spends about 30% of the healthcare expenses. These are diseases like pulmonary disease and cancer and trauma, a lot of heart disease, and certainly a lot of mental disorders. About 5% of the population accounts for almost half of the U.S. healthcare population expenses. So it's the idea that going after certain high-risk patients or really hotspotting can have a significant impact on the healthcare system. So I got this question at the seminar where I gave this talk and actually went back and figured out the economics of some of the things that we've done with this single family. And it turned out that just for this initial 30 days in the hospital, in our heart hospital, it cost this family about $1.2 million, or at least the healthcare system about $1.2 million to take care of this patient. Compare that to what is about a $238, $231 cost to give this drug combination to this patient over time. And in fact, we've now figured out that the cost for these 20 plus events that this patient has had over the last 2 years, and it's well over $15 million, unfortunately. So the idea is that if we could find these patients over time, that we could have a big impact on the healthcare system. The question is, can we use this to prevent future family events? So this is one where I show a slide, but I think it's careful that we think a lot about genetics before we broadly say that this is the smoking gun. So again, you can see the father and the son here, where you can see the variant and the specific gene on the top and no variant on the bottom. The question is what do you do with a patient like this? What do you do with a family like this? Is this enough? You know, we talk about being able to use this to predict arrhythmia events 30 years in advance. Is this story enough to actually do that? And I would argue probably not, and this is why we're still working on this. There's a lot of diseases where a single gene hit, like cystic fibrosis, we know can cause severe forms of disease, and it's black and white. In this sort of case, I would argue that we still know simply not enough about these molecular pathways to be able to make these diagnoses and treatments. So this is something that I think, a word to the wise, that we have to be careful when we're interpreting our data to make sure that we don't over-interpret it. Okay, so this really brings us to sort of the guts of our program, where we're really looking at, like Yoram said at the start, new mechanisms for arrhythmias that focus around ion channels, but certainly are not just focused on the ion channels themselves. We've known a lot about channelopathies due to a lot of work of people in the room today. We know that mutations in ion channels and transporters certainly have a big impact on cardiac rhythm, excitation and traction coupling, but just as important, regulation of communication between neurons, regulation of salt and water balance in the kidney and the gut, on and on and on. We know that mutations in these obviously can cause severe forms of things like long QT syndrome, a lot of neurological diseases, as well as things like cystic fibrosis that I mentioned before. Our lab, as Yoram mentioned, has been focused a lot on these proteins that are called ankyrin for the last 12 or 15 years. These are proteins that were originally identified in the red blood cell as a critical linker protein between the actin and spectrin-based cytoskeleton and membrane proteins like the N3. So when I explain this to graduate students, the best way that I can imagine is that when you think about an ion channel, think about it as a buoy moving in the water, and the buoy is not effective in marking the channel unless it's actually attached to the bottom of the ocean. And so thinking about the ankyrin of the spectrin, that's this chain that links to the bottom of the ocean. As a lot of you know, not only do we teach graduate students, but we also teach medical students and surgeons, and so this is the analogy that I use for that, where we think of these as the ropes that you use to tie your yachts off. No? All right. So a lot of work has been done over the last 30 or 40 years now, actually being able to understand what these ankyrin do in human physiology. A lot of this work goes back to sort of slaughterhouse biochemistry, where, again, a lot of kidneys and hearts and brains really were purified ankyrin binding partners. And a lot of the proteins that are absolutely critical to cardiac physiology or neuronal physiology are on this list. So things like voltage-gated sodium channels, sodium-potassium ATPase, sodium-calcium exchanger, Colin's not here, but ATP-sensitive potassium channels. So this is sort of one class that ankyrin have been really critically involved in. What we've learned, though, most importantly, I think in the last 5 or 10 years, is that they're not simply ion channel associated proteins, that they really have a big role in regulating cell adhesion. They associate with a lot of molecules that have, say, rogue F activity And then they also bring a lot of other proteins to the plasma membrane, so things like protein phosphatases, protein kinases, things like clathrin, molecules that are involved in movement of organelles along microtubule pathways. So why do we study ankyrin? Well, almost originally immediately after ankyrin were identified, Van Binnen and Peter Agre and colleagues learned that mutations in ankyrin, specifically in erythrocytes, could cause severe forms of hereditary spherocytosis. So we know that this beautiful sort of membrane structure in the erythrocyte could be compromised by mutations in both ankyrin or spectrin that cause these basically the membrane to collapse and give you these spherocytes. The other reason that we've really gotten interested in ankyrin is that at least in metazoan cells, so non-yeast cells, we've learned that these proteins really have a big role in getting proteins from point A to point B in very elegant and very complex tissues. So things like the red blood cell, but also cells in the central nervous system like lung axon tracts. So the question that we asked is this true in other cells, and dysfunction in these other tissues, does it cause phenotypes? So one of the big areas that we've studied over the last 10 years is the role of ankyrin in regulation of voltage-gated sodium channels. And I know Tom Hund was here a couple of months ago, so I'm not going to repeat a lot of this work, but in fact that we've learned that ankyrin-G, which is ankyrin-3 and also found in the brain, regulates sodium channels, and we've learned that at least at the intercalated disc, ankyrin-G directly associates with very specific regions of NAV1.5, to where that if you lose ankyrin in a cell, you lose big components of voltage-gated sodium channels. We've also found mutations, and specifically in Brugada patients of NAV1.5, that affect this interaction with ankyrin-G that cause severe forms of the Brugada syndrome and potentially sudden cardiac death. So again, ankyrin-G, which is ankyrin-3, is required for voltage-gated sodium channel trafficking, and dysfunction in this leads to severe forms of sodium channel defects in Brugada syndrome. The other ankyrin that we're actually interested in is an ankyrin called Ankyrin-B. So Ankyrin-B was first associated potentially with long QT syndrome by a very good group of colleagues in France, associated with a family that had a severe form of long QT. This is called long QT type 4, where you have reasonably normal EKGs at rest, but then following exercise or catecholaminergic stress, severe forms of ventricular fibrillation and potentially sudden cardiac death. So we hypothesized, based on the linkage of ankyrin to this general region where it was mapped, where this disease was mapped, that this might be the potential causal gene of this disease. Unfortunately, as a lot of you know, that everyone that has a favorite gene and there's a locus, you imagine that your gene is the one. And when we started looking at the genetics, it turned out that there were 18 other candidate genes in this region that were a lot more attractive. There were things like CAMP kinase was in this region, there were trip channels, a lot of other proteins that you might be more liable to think for a CPVT type of disease. The other reason, at least at this point in the literature, is that all of the other long QT genes were either ion channels or very appropriately ion channel regulatory proteins. So we knew that long QT4 was a dominant form of arrhythmia, and that the idea would be, if this was ankyrin might be the disease candidate gene, that the ankyrin-B heterozygote cells, so cells that had half as much ankyrin, might show phenotypes. So we started doing experiments with mice, and this is technology that's very familiar with most of the people in the audience. Instead of just looking at subsurface EKGs, you put telemeters in, you can run the mice, you can stress the mice, you can give them injections and look at their EKGs. At baseline, when we did this, we were quite disappointed and didn't see a lot of huge impact of the loss of half of the ankyrin on the cardiac EKG. However, when we started exercising the mice or giving the mice catecholaminergic stress with isoproterenol or epinephrine, you can see that we were able to very nicely reproduce a lot of the phenotypes that we found in the patients. And in fact, a lot of the mice would actually undergo these runs and suddenly drop dead. So this was our idea of a potential model for the human long QT4 gene, and I'm not going to go into all the details, but we've gone on and done a lot of imaging. We've done a lot of looking at things like serum potassium, and in fact could show that they had a lot of the very same characteristics of the patients that in the original long QT4 kindred. So we've gone on to be able to use big pedigrees that had ankyrin mutations, and we're able to show that in fact there was a single mutation in this large fourth generation French pedigree, all that were located to blue 1425G, which is in the C-terminal domain of ankyrin. So this is not in the part of the ankyrin that binds to the ion channels, but the part of the ankyrin which basically regulates its catalytic activity. We've gone on now to identify 9 different loss of function mutations in the ankyrin-2 gene, and in fact we've now been able to put together a screen for variants where these patients can be put on the appropriate medical therapy or have surgical therapy. So the question though still from a sort of fundamental cell biology aspect was what's ankyrin actually doing in these cells? We knew it was involved in the disease, we knew that it was causal for forms of potential ventricular fibrillation, but really what's it doing? And when you put it in context to some of the other genes, it didn't really make a lot of sense of how it fit into the normal paradigm of ion channels. So again, using a lot of different techniques, this is experiments where we went back to the slaughterhouse, got a lot of tissues, and were able to actually purify ankyrin with a bunch of very select ion channels and transporters. We're able to show that ankyrin actually binds to a very select group. This is ankyrin-B, a very select group of cardiac membrane proteins. One is the sodium-calcium exchanger, the second is the sodium-potassium ATPase, and the third is the IP3 receptor. These are very high affinity interactions, so if you do scattered analysis, you can see that these are very, again, lock and key interactions at the level of about between 5 and about 15 nanomolar. So again, what's happening in actual real cells? So we went back to neonatal cardiomyocytes, which again are a little bit easier to image. It allows you to transfect the cells to be able to do rescue assays. On the top, it shows you what ankyrin looks like in a neonatal cardiomyocyte at about day 7. And you can see in these myocytes, we start to see sort of the nascent striations of the sodium-calcium exchanger in these cells. I think this image on the bottom really nicely shows what we see when we lose ankyrin. So ankyrin, when you see it at certain parts of the cell, the sodium-calcium exchanger is there. When you don't see ankyrin-B in certain parts of the cell, you see a complete loss of the sodium-calcium exchanger. Some of the things that we're still trying to figure out is why do we only see half as much ankyrin, not throughout the cell, but in specific regions of the cell? And we have new data now that it looks like that ankyrin actually is involved in nucleating key membrane sites to where things get recruited to it. So if you lose ankyrin itself, you actually lose these nucleation sites. So again, the next question is, does loss of ankyrin-B function result in electrical abnormalities, or are we simply losing the structure of the cells? And so these are experiments that were done about 9 years ago, but actually really best implicate what we saw. Again, these are neonatal cells, so again, I understand that there's a lot of difference between these and adult cells. This is a neonatal cell culture from a wild-type mouse, and compare that with a neonatal cell from a heterozygote cell. So 2 things. One, you see obviously changes in them, the frequency of contractility, but also the way the cells really start to shudder and shake in certain regions that nicely correlates with this picture I showed before. So when we look at calcium, which obviously underlies this contractility, you can see, not surprisingly, that there's big difference in the way the calcium is handling, even at the level of a neonatal cardiomyocyte. So again, on the left, you see very nice rhythmic calcium waves that spread across the cell. Compare that to a heterozygote cell, and you can see that the cell, again, not only the frequency goes down, but we also see this sort of stopping and stuttering before the cell freezes, and then it'll eventually contract again. So that was in neonatal cells. We've gone on now to do this in adult cells, which is a little bit more quantitative. We can actually look at calcium transients and calcium sparks. And just to show you some of the data that we've seen, that we see looking at these cells and looking at calcium transients, we see bigger changes in the heterozygote than we do in the wild type. And then very similar to what we see not only in the mice, but what we see in the patients is that at rest, at baseline, we don't see real changes in the axe potential. So surprisingly, we didn't see huge changes at baseline, but then similar to what we saw in the patients, similar to what we saw in the animals, you give a little bit of isoproterenol, you give a little bit of stress, and we see very pronounced extrasystoles, which are obviously the cellular correlate of an arrhythmia. So this is our model now for what we think underlies Ankyrin-B syndrome. So just like giving toxic doses of digitalis, which actually in a normal case could increase cardiac contraction, that loss of Ankyrin-B has two hits. One on the activity and the levels of the sodium potassium ATPase, and secondly, a second hit on the sodium calcium exchanger. Both of these, again, if in the right dose, could actually increase calcium transients, increase contractility, just like digitalis. However, if you give a little bit too much or decrease ankyrin too much, then this obviously can cause extrasystoles following catecholamine stimulation. So to summarize the second part of the talk, that the molecular basis of this disease called LONQ-T4, which we now call Ankyrin-B syndrome, which is probably a little bit more like a CPVT than a LONQ-T syndrome, is a mutation in ankyrin. Ankyrin targets 3 functionally related proteins that all have a major role in regulating intracellular sodium and calcium. And then we've learned that ankyrin dysregulation very nicely mimics the activity of digitalis in the heart. The hearts beat a little bit stronger, but they're much more prone to having cardiac arrhythmia. Some of the things that we're working on now in the lab now are taking this a little bit further to look at how does environment affect ankyrin interactions and how does environment affect the progression of disease. And this is one patient that we're working on now where that had an ankyrin mutation but refused to take treatment, and in fact was a marathoner. And so we've been able to learn that over time, in fact over decades of exercise, that these patients actually with high levels of calcium can begin to remodel. And in this case, a very kind of boring run-of-the-mill sinus bradycardia and CBAC type of phenotype can progress to a severe form of ARVC in these patients if untreated. So the question is, what about beyond the ventricle? Everything I've told you about before is very focused on sodium channels and sodium potassium ATPase and sodium calcium exchanger. What about other regions of the heart? And this is work that's a little bit more recent in the lab on new and unexpected roles of ankyrin. So again, going back to families, this is a second French family. It's nice to work with French families to get to travel to other fun parts of the world that all also have ankyrin mutations, but also have some other non-ventricular phenotypes. These are patients that all have severe sinus node dysfunction, and the penetrance, unlike LONQ-T or Brugada, which is maybe 10 or 20 or 30%, the penetrance in these families is about 90%. So if you have an ankyrin mutation in these families, you're going to have sinus rates in the high 30s or the low 40s and requiring a pacemaker. So this is one of the families that we've looked at. This is an EKG of a 7-year-old girl on the bottom, and you can see not only do we see a very low heart rate's in the 30s, but we also see heart rate variability. So you see an arrhythmic on the bottom, a long beat, a short beat, a long beat. So this is not simply on the course of looking at hours or days, but really on a beat-to-beat basis, we see changes in automaticity. Questions were, is this something that's happening due to development? Are we seeing these actually hardwired in these children? And we can actually go back to 32 weeks for the same little girl, 32 weeks after conception, and you can see a normal heart rate of a little girl this age should be about 150 beats per minute. This little girl had a heart rate in utero of about 86 beats per minute. So these are really hardwired defects in these kids. Question is are we looking at single families, or are we seeing this reproduced with different variants in different parts of the world? This is actually a second family where we can do good genetic analysis and look at log scores where we can do tracking. And again, an 8-year-old little boy has the same, very similar phenotypes. Again, a very low heart rate, but you also see this heart rate variability in these children. So really questions line up very nicely. What is the molecular defect? Is this a sinus node defect? We also obviously know that sinus regulation is involved not only in the heart but the brain. Are there neural input defects? And in fact, is Ankyrin-B even expressed in the sinus node? So this is where it's nice again to have human tissues like you guys use here at WashU, where we can map the hearts. We can actually go in and carve out the central sinus node and be able to look to see whether there are significant levels of Ankyrin-B. And again, we can do very similar to what you guys have done here with positive and negative markers. We can show that pretty much there's Ankyrin-B everywhere in the sinus node region. So some of the tools we've been able to use would be mice to be able to actually go back and look to see what are the potential causes of sinus node defects in these animals. Is it that because they have ion channel defects? Is it because the sinus node doesn't form correctly? Is it because there's issues with block out of the central sinus node? And so these are just images looking at a right atria from a mouse that has all of its ankyrin here in green, or half of its ankyrin here on the bottom. And you can see that looking at HCN4 staining, which is the marker for the IF current, that there are plenty of cells in the sinus node of these animals, but we simply see a decrease in the expression of the ankyrin-B. So again, this is great to look at immunofluorescence, but a lot of us actually want to see functional phenotypes, and so we started looking back again at our animals to see, certainly, do they have sinus node issues like we would see in the patients. And so again, this is about 12-minute traces from about 10 mice that are the wild type on the top in purple, and then red for the heterozygote. And again, it's very consistent where not only do they have heart rate loss, but extreme heart rate variability on really a beat-to-beat basis, which is still surprising to me. So again, the question was what is the molecular defect underlying some of these changes? So we've gone back and started looking at using electrophysiology and immunostaining and RNA-seq, like a lot of you use, to figure out what are some of the differences. We thought there might be changes in the sodium-potassium ATPase and the sodium-calcium exchanger, just like we saw in the ventricle. But in fact, how we now do control experiments in the lab is that we again see things that we didn't expect to see. So on the top, let me take you through this. This is staining for HCN4, CAV 1.2, CAV 3.1, and CAV 1.3 in a wild type sinus node cell. So again, single sinus node cells, which is one slide, but it took the postdoc about 53 years to learn how to do this. So on the bottom, you see cells from the Ankyrin-B heterozygote mice, and again, no big difference between HCN4, no big difference between CAV 1.2, no big difference between CAV 3.1, but when you look at CAV 1.3, which is another form of the L-type calcium channel in these cells, we see dramatic changes in its distribution in the cell. So instead of being around the edge of the cell, like its sort of close colleague CAV 1.2, we can see most of this found in early endosomes in these cells never able to reach the plasma membrane. So that, again, a lot of us that use immunostaining, that everyone can always find a representative image. So we actually also go back and prove this with electrophysiology and can show, similar to what we showed with the immunofluorescence, that T-type channels are just fine in these cells. We see no problem with IF current. But when we start looking at the components of the L-type current, we see about a 50% decrease in the L-type current, which is encoded by CAV 1.3. It has a little bit different biophysical properties, and we can actually isolate these out. So when we start looking at calcium, again, it very nicely mimics what we see in the patients. It very nicely mimics what we see in the isolated parts, to where at baseline, unlike the wild type, that again, that has very, very regular calcium waves and calcium sparks. When we look at this in explants or even single sinus node cells, look at calcium transients or action potentials, we see not only a decrease in rate at the start, but again, very similar to the patients, it goes through this sort of tachybrady phenotypes. And we've been able to actually trace this back to CAV 1.3 in regulation of what we call the diastolic depolarization rate. Okay, so to sum up the third part of the talk, we've learned that ankyrin-2 variants are the first large-scale genetic link to human sinus node dysfunction, and probably the most penetrant form of arrhythmia that's been identified in the patient population. Humans with ankyrin-2 variants display sinus bradycardia and extreme heart rate variability. Yeah.
So since there's really little T-tubules in the sinus node, and your hypothesis in the ventricle is that it's kind of organizing T-tubules, do you think they're just more sensitive there because there's less, why is it affecting the sinus node and not the ventricle?
Yes. Yeah, let me finish that, okay. And I'll come back to that. So yeah, so we've learned that ankyrin-2 variants are the first large-scale genetic link. That humans that have this not only have bradycardia due to the CAV 1.3, but there's probably some other changes, and I've not talked about it today, but ankyrin, we've learned, has a big role in targeting some of the subunits that regulate IKCH, that regulates sympathetic, parasympathetic regulation in the heart. Mice with reduced Ankyrin-B really nicely mimic some of the phenotypes that we see in the patients. and we've been able to see that single sinus node cells display abnormal electrical activity. One of the things that was very surprising to us in these studies was what you just brought up, is that when we think about this cell membrane of the sinus node, we think of it as again, obviously very different than the ventricular cell. There's no T-tubules. But what this is telling us is there's probably key microdomains for CAV 1.2 and 1.3, even within a sinus node. And so very likely there's also going to be the same version of what's happening on the SR as well. So again, to us, this was extremely surprising. Amy Lee has some data as well that shows similar results, that they're very likely different microdomains of 1.3 and 1.2. We've actually mapped the binding site of ankyrin on the CAV 1.3, not distal C-terminus, but sort of in the last cytoplasmic domain. But what it's regulating and how it's doing that is a big question. So in these cells, we think that this is probably regulating trafficking or at the level of the late endosome to get to the membrane, and less a role in retaining it at the membrane. And we've got some pulse J's to show that. Okay, so again, the final thing is dysfunction in this CAV 1.3 has a big role, not in the normal calcium release, but probably more in the diastolic depolarization region for these sinus node cells. So just to quickly summarize the talk, we've learned that ankyrin-G targets very critical voltage-gated sodium channels in heart cells, very similar to what it's doing in neurons. The human SCN5A variants that affect the interaction between ankyrin-G and NAV 1.5 lead to decreased NAV 1.5 at the plasma membrane and can cause forms of Brugada syndrome. We've also learned, and probably Dr. Hun talked about this when he was here, that this complex brings in other regulatory proteins to the sodium channel complex, including things like PP2A, as well as kinase that have major implications for sodium channel late current regulation, and that's an active role of one of our grants now as well. Ankyrin-B targets the sodium potassium ATPase and the sodium calcium exchanger. It also targets the IP3 receptor, which we're still trying to figure out what its role is in the ventricle. But we do know that ankyrin-2 variants causing ankyrin-B loss of function lead to issues with calcium homeostasis, leading to phenotypes that nicely mimic the activity of digitalis. One of the exciting things that we're working on, in addition to some of the epigenetics of these diseases, is looking at some of the other phenotypes that these patients have. So again, starting with an EKG where you can have a mutation and clearly show a phenotype, like what are the other things that ankyrin's doing? And we have pretty good data now that ankyrin is involved in certain forms of bipolar disease, and ankyrin-G is involved in certain forms of schizophrenia, which has been obviously a big question in the literature. There's been a lot of GWAS that have associated these proteins with these diseases, but not a lot of cause and effect. Okay, so the third part of the talk, we talked a little bit about how ankyrin-2 variants of the cause of human sinus node disease, and how ankyrin targets very specific forms of calcium channels important in regulating sinus node diastolic polarization. So again, whether you're an ankyrin aficionado or probably not, hopefully this has convinced you that simply getting an ion channel to a plasma membrane is a little bit more complicated than we used to think, and that certainly location of these proteins, not only to the plasma membrane, but relative to other molecules, whether it's a signaling molecule or in fact another ion channel, are absolutely critical. So what's next in our lab? Well, certainly one of the things that we always start with is patient populations. This is a new patient population we've been working with for the last couple of years. This is a really interesting Indian tribe that's located about 9 hours north of Vancouver. It's an Indian tribe called the Jikson population. They have about 5,400 members, and for as long as anyone can show in their written history, they've had severe forms of arrhythmia and sudden cardiac death. A few years ago, they were shown to have some potassium channel mutations that explained about half of the tribe's phenotypes, but the other half had gone unsolved for the past few years. We've been able to work with some of the physicians and genetic counselors in this region, and actually found that a lot of these patients do have ankyrin phenotypes and ankyrin mutations. What's interesting though is they have a number of other phenotypes that we didn't expect, including aneurysms, and this is allowing us to get into some of the roles of ankyrin in cells that we really didn't have a big understanding of before. So this is where we'll end today, and hopefully the next time I'm here, I can tell you some new answers about some of these populations. So with that, a lot of collaborators and a lot of very helpful mentors that helped us put this story together. A lot of the people that we work with at Ohio State, but importantly, a lot of physicians and clinicians and electrophysiologists that have shared, been very gracious in sharing a lot of their patients with us. And with that, I will stop and take any questions. Thanks.
Questions, comments to Peter?
In your adult ankyrin heads, do you see that spotty immunohistochemistry pattern that you were seeing in the neonatal?
Yeah, so the question is you know, I showed you these pictures of the neonatal cells where there's parts of the cell that have well-established T-tubules and other parts that don't, and the answer is yes, not to the same extent that we see in our neonatal cells. What's interesting is when we go back and try to rescue some of the phenotypes in the neonatal cells, we can restore T-tubule localization to these. When we do this in the adult cell, obviously things are a little bit more differentiated, and so it's a little bit harder to. So the answer is yes, but not to the level that we would see in the neonatal cell. Certainly enough to create heterogeneity in calcium management. So that's something that we're working on now. Please.
So it looked like you had some really interesting dynamics in your pacemaker with the SA so like you had slow beats and then fast.
Right.
There were periods. I was wondering if you had thought about putting that in a mathematical model to actually figure out how it's entering and exiting those regimes with the calcium.
Right. Yeah. So when you look at this, you know, the first thing that I think a lot of us think about is that that it's slow. You load the SR and it's bang 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 bang. Certainly that CAV 1.3 can probably answer some of the issues about basal rate, but probably not some of that loading in rapid firing. We're working with Tom, obviously, to do some of the mathematical modeling. A lot of it we think is related to movement, like was brought up earlier, of how the release sites are associated with the stores. And certainly we have some of the data with actually a whole organ where probably some of these Kirk channels that may have a big impact on sort of how that firing is regulated.
In the ankyrin heads, the first question is, does the ankyrin dimerize, and is the head right? Does it require dimerization in order to have a homogeneous effect around the cell, and is it concentration dependent?
Yes and yes. Ankyrins sort of are, normally look like this when we show them on a PowerPoint slide, but it turns out, in fact, that ankyrins can bind to themselves to regulate their activity. They can be in closed form, which is inactive, and open form, which is active. When this happens, obviously, you can have, they can dimerize and on head to tail, and just similar to how spectrins do as well. And so a lot of our thoughts of what these human mutations do is actually act as dominant negatives for the wild type alleles. And so simply having half as much of a very abundant protein, we would think that wouldn't be as severe, but certainly it appears it is. The second part of the question
did that account for the localization that they couldn't dimerize?
Yeah, it probably has probably not in the hets, but certainly with the human disease, we think that probably plays a bigger role. That in a human, having a normal allele and a mutant allele is probably worse than having half as much ankyrin.
If you do a pull down with ankyrin, how many different things do you see? Not, like half to 90% of them will be irrelevant, but.
Yeah, I'd say more than that. So the problem is, as soon as you start doing pull downs, these are pretty insoluble proteins, and as soon as you start to get to the point where you can solubilize these, you start bringing down actin and microtubules, and at that point, I think most of us
once
that
the
do biochemistry
microtubules go down, everything.
Well, I think most of us, once you start pulling down the kitchen sink, then most of us would probably argue that it's a crappy experiment. And so what we've tried to do is, and I've tried to show you, is to take these unbiased approaches where we look at the active potential and do modeling, or we look at basically libraries of ion channels, or we've done yeast 2 hybrid a lot to look at, that's a great way as well, right? But to look at proteins that might be changed, and then immediately go to the animal models to say, do we see loss of these? We've been able to, over the last 10 years, accumulate a pretty good biobank of hearts or explanted tissue from patients that have ankyrin mutations. So we can actually use those as well. Obviously, iPS cells is another way that this can be done, but our lab has probably not gotten as good in that as we should probably need to.
Yeah, our lab use Xenon oocyte and molecular to study ICAS channel. Do they have ankyrin in the Xenon oocyte or in mammary cell?
Yeah, so everything has some form of an ankyrin, you know, and again, depending on how far you go back, then there's certain forms. Drosophila have ankyrin, you know, Xenopus have ankyrin. You know, it's just which version or which forms of ankyrin do they have.
So do you have knowledge whether ICAS channel or KC1 channel associated with any kind of ankyrin?
Right, so we do know that KC&Q2 and 3 have ankyrin binding sequences, and so those are encode current and more relevant in brain. The type 1 does not have an ankyrin binding sequence, and we've looked at that in detail based on a lot of reviewers' questions about that, and we've not been able to see an interaction. It turned out that it's because they don't have the binding site.
Okay, thank you.
Is the tachycardia in your tachybrady, is it sinus tach, or is it like a fib flutter kind of thing?
It's like a fib, more of a fib
Okay.
flutter, yeah.
So then maybe that's less of a modeling and more of a, you know, because it's not really the sinus, then it's really,
Right.
well, maybe.
Yeah, so I thought we kind of had a decent idea of what was happening until we started getting more and more. Again, the more patient, we were starting out with a pretty small N of we've got a lot of mice, but very few humans. By starting to get some of the data over time, again, we never would have predicted we would start seeing ARVC phenotypes, and so to sort of guess what some of the phenotypes are versus what's happening with the environment has been interesting. And again, we just need more and more patients to be able to try to.
Have you looked at the atrial myocardium?
We have, and in fact, some of the same. We sort of see a blend of what you see in the sinus nodes. So we do see changes in CFE 1.3. We do see changes in the exchanger. We do see changes in the pump. Patients that do start out in sinus bradycardia and sort of this tachybrady syndrome then progress to atrial fibrillation and pretty severe forms of AFib. And so it's, you know, we identify these patients at kind of 2 or 3. They develop sinus bradycardia by 3 days of age, and then it progresses into an AFib. So it's a pretty interesting disease because what you see at day 1 is very different than you see at literally day 30 or 40 in these kids.
And the mice, though, did they get the fib flutter?
They do, yeah.
okay.
So obviously AFib in a mouse is...
Right.
Wow. We can, you know, there's ways that you can go in and
Yeah.
and we can, you know,
Stimulate and
you
make
can
it
stimulate
happen.
them, and you know, that's whatever you believe in that sort of data. But we can. What does it mean? Yeah, not a lot. So the kids have it though, which is probably the more important question. Gene.
So why don't people who have all these variants have many more neurological disorders than they display, I should say? Because, I mean, your DPP6 variant, I assume it's expressed in brain tissue?
It is, and in fact, that's the reason he's not a transplant candidate, is that there are severe bipolar issues in this gentleman. And so, you know, lithium dependent, and in fact, what's cool about this, and I didn't have time to talk about it, is that the sort of the combination therapy has a big impact on his emotional state, which is interesting. Again, this DPP6, I still have questions about, you know, how much there actually is in the human heart, and you know, the issue with the time dependence, particularly when you start talking about someone in their 30s and 40s, I still don't get that part of the story. And you know, I've talked to Arthur a little bit on his family members and some of these other families in the Netherlands, and he doesn't know as well. It just doesn't make sense from sort of an evolutionary standpoint.
But that's the age group that begins to get psychiatric diseases like bipolar and schizophrenia.
Agree. This patient had psychiatric disorders when he was 8 years old. So,
Well, of course there are exceptions, but it's the
Certainly.
overwhelming amount for
Yeah,
teens and
absolutely.
20s and everything.
But isn't it also localized to certain areas of the heart, like the Purkinje's cells?
right. So there's been some experiments that, like the Natal group has data that this DPP6 is highly expressed in Purkinje's, and some of the patients from the Netherlands group have been ablated into Purkinje, and that solved it. This didn't work in this individual, and what we've seen is, when you do RNA, that you can find DPP6 everywhere, but there's not a lot of it, and when it's enriched in something where there's twice as much of not very much, it's sort of what do you make of that physiologically? So that's something that we're trying to do.
And if you compare it to the expression levels in the nervous system, it's 100-fold
Oh,
higher
at least.
everywhere.
Yeah.
In the nervous system?
Yeah.
So like the CT values are a couple log orders.
Yeah, it's one of the
So you
most-
think it's an autonomic problem, maybe in like the VT?
So I don't know, but you might even expect that they have lots of problems. Epilepsy,
Sure.
motor dysfunction. I mean, all kinds
Well-
of things.
well, these
or
are
pain problems, right? Pain perception. I mean, it's quite interesting.
syncope patients, right? It's like they go to the neurologist. It's not the
Sure.
brain, and then they come back to the cardiologist. Not the heart, not the brain, not the heart. We can't. Yeah. So they just have kind of this weird,
Yeah,
passing
and
out.
I
And
think in this patient it was what's the most severe phenotype that day, and in this case, he'd had on the first story he'd had neurological phenotypes his whole life that were controlled, and then his heart is now fine, and now it's sort of going back to understanding his neurological phenotypes.
So
But
is there any KB4 gain of function mutation that increase ITO by those mutations? Do they have similar phenotype mutations, or is there any?
Not like this. No. You mean in the heart?
Yeah.
No.
Yeah, there's only been one identified gain of function in Brugada. And it's
It's-
one person,
yeah.
no family. I don't know whether anybody believes that it's really.
Yeah.
So so the phenotype is not just maybe not just caused by the increase of ITO, something else is also.
We would guess that that this DPP6, in addition to binding to this specific potassium channel, probably has roles in regulating other potassium channels. I don't think that's surprising to anyone in this audience. I think any of us that have used 4AP in the lab or have read more than 2 papers ever know that this is an incredibly dirty drug and can block a lot of K channels. And so that's why at the end of that part of the talk, I want to be careful that we make sure we understand that some of the sort of the antiarrhythmic effects may be a general sort of dulling of multiple K currents, which a lot of folks have hypothesized for a long time could be potentially antiarrhythmic. If you give them to the wrong patients, obviously it could be pro-arrhythmic. And so I think it's being careful in these patients to kind of know what you're dealing with.
Yeah, so one of the things that actually occurs, it strikes that you study a lot of the proteins that are promiscuous to many different channels, and when they get, have mutations, what is the general way that people try to figure out, what's the real target that these mutations actually?
Yeah. So it's a good question. So what we would always do is we go back to animals, or we go back to the human sections. That if you study a lot of these interactions in a test tube, or you study them by a yeast-2 hybrid system, a lot of these proteins can all interact, and it's no different than most of the other proteins that we all study in our lives. But what we've learned is that in real cells, in real animals, or in patients, that all of these have very specific zip codes. So while I talked about ankyrin-2 and ankyrin-3, or ankyrin-B and ankyrin-G, they look very similar, and their structures are similar, and they catalytically behave very similar. They each bind to different groups of proteins. So ankyrin-G in the heart is very specific for voltage-gated sodium channels and can't bind to the exchanger of the pump, whereas ankyrin-B is the exact opposite. And we've mapped specific binding sites on either, where we can do site-directed immunogenesis and knock them off. those affinities are incredibly high. If you did it though in a test tube with a pull-down assay, they're all gonna dissociate. So we would be very adamant that these are interactions that need to be confirmed in vivo, and that simply looking at them in a test tube is a good start, but probably not the way to go.
Back to the DPP6. When you express it in heterologous cells, you got a significantly more current with the T versus the other one of the other splice variants. In the heart, in the human heart, do you think it's that the 6T is being overexpressed to what the low level, if it's there at all, or do you think it's that by removing part of that C terminus, you're almost like leaving a break on ITO?
Good question. I think in some with data and sort of a lot of sort of gnashing my teeth about some of the results we've shown is I don't think there's a lot of the canonical form of baseline, and I think this small version expresses well. It probably is made, it's made better, it's expressed better, it may be more stable. So I think there's some other things that are happening here. I think that when we look at, again, at basal levels of DPP6 to begin with, there's not a lot. So I think that having a version that may be regulated over development, where 6T comes on later and it becomes sort of the DPP6 that we've all been looking for. I think there's a whole lot of the 6 in brain, and probably that's what it's doing there. All right.
Okay,
Thank you for your questions.
any other questions? Thank you for a great presentation.